Science e-Solution. I am Manju Sharma here today again with another video tutorial. Today's video we are going to learn about an experiment which is going to prove that chlorophyll is necessary for photosynthesis. We all know plants perform photosynthesis for synthesizing their own food, and plant can perform this function because they are having a pigment called as chlorophyll. So in this experiment, we will show how it is proved that chlorophyll is necessary for photosynthesis. We'll prove this statement in 6 steps. So there are 6 steps for this experiment. In first step, we will take a potted plant with variegated leaves. So we are going to take a potted plant, and the plant of which type? Plant which is having variegated leaves. Now what are variegated leaves? Variegated leaves are the leaves which are having 2 zones. At least 2 zones should be there. One zone should have green color, or the other zone will be having another color. For example, white and green, 2 colors are there in the leaf. Generally we see the leaves of green color only, but variegated leaves are the leaves which are having more than 1 color. So in this experiment we will be taking a leaf which is having 2 zones. One is green color and the another one is white color. So one leaf having 2 colors, that is called as variegated leaves. So first step, take a potted plant with variegated leaves. Now why we are taking variegated leaves? Because green color contains chlorophyll. That means it is clear from this experiment. We are taking a leaf which are having 2 zones. One is green and the another one is white. So the portion which is green, it is having chlorophyll, and the portion which is not green or which is white, it is not having chlorophyll. So it's like control of the experiment. Now second step. Keep this plant in a dark room for 3 to 4 days to de-starch its leaves. What we'll be doing with this plant? We'll keep it in a complete dark room so that no sunlight can enter in that room. So we will keep this potted plant in a dark room for 3 to 4 days. Now why it is done? Because you see, de-starch its leaves. Plants prepare their food by photosynthesis and store it in leaves in the form of starch. Means they prepare glucose and the glucose is instantly converted into starch. Now when we are keeping this plant in a very dark room for 3 to 4 days, all the starch which has been synthesized previously by photosynthesis, it has been used. That means the leaves are now de-starched. They have used up all the stored starch in their leaves. So by keeping it in 3 to 4 days in a dark room, we have de-starched its leaves. Now no more starch is present. Correct? Then in the third step, we'll take out this potted plant in sunlight and keep it for 3 to 4 hours. You can also keep it for 5 to 6 hours. Now after the leaves are completely de-starched, we'll again take the pot and keep it in light. That means under the sunlight, so that photosynthesis could take place for those 5 to 6 hours. Now you see, when we put the plant in sunlight, photosynthesis takes place and again it starts to produce its own food, that is again starch is produced. Then the fourth step, break one leaf of the plant and boil it in alcohol. Now after 5 to 6 hours, what we'll do, we'll take out, pluck one leaf, put it inside the test tube in alcohol and then put this test tube in a hot water bath. This is how we boil alcohol. So we will break one leaf, pluck one leaf of the plant and boil it in alcohol. Now boiling in alcohol, why do we boil in alcohol? Because alcohol removes all the chlorophyll which is present in the leaves. Now we'll be removing all the chlorophyll which is present in the leaf by boiling it in alcohol, which is the fourth step. Fifth step, we'll wash the leaf with hot water. After boiling, we'll take out the leaf and wash it in properly in hot water so that if any chlorophyll is left over in the leaf or is sticking to the leaf, it gets removed by washing with hot water. Clear? Now, till now what we have done? We have taken potted plant with variegated leaves. This is very important. Then we have kept this plant in dark room for 3 to 4 days so that all the starch which has been prepared is used up. Leaves are now completely destarched. Okay. Then we'll take out this potted plant in sunlight for some hours, 5 to 6 hours, so that it could perform photosynthesis and it could synthesize its own food. Okay. Then we will pluck one leaf, boil it in alcohol to remove the chlorophyll. Again the leaf will be washed with hot water so that chlorophyll, if any is present, is removed. Clear? Last step. We'll put iodine drops over the decolorized leaf. Okay? The leaf which is decolorized, that means whose chlorophyll is removed. Ultimately, after removing the chlorophyll, the leaf will be decolorized. So, in this decolorized leaf, we will put iodine drops. Now, what is the sense of putting iodine drops? We all know iodine is an indicator of starch. That means if we put iodine drops in a solution which contains starch, the solution turns blue-black. And if the solution doesn't turn blue-black, means there is no starch present. Clear? Last step will be putting iodine drops to see whether the starch is present or not. Okay. Now, what do we observe? After putting iodine, there will be some observation. What do we observe? That the white part of the leaf, that means the part of the leaf where no chlorophyll was present, which was white part, it turns blue-black. It does not turn, white part of the leaf does not turn blue-black, indicating no starch was formed in this part. White part, it doesn't contain chlorophyll. It cannot synthesize starch. Okay, that is why starch is not formed, and that is why it does not turn blue-black. Okay, white part does not turn blue-black. Now, the part of the leaf which was green in color initially, because after we have washed with alcohol, washed with hot water, all the color and pigments are gone. So, initially the part which was green, it will be turning blue-black after we put iodine, because starch is formed in this part. Clear? When we have kept the leaf to perform photosynthesis in sunlight, starch is formed. So this is the observation. The part which was white, it doesn't turn blue-black, showing no starch was formed. The part which was green, which was having chlorophyll, it turns blue-black, showing starch was formed because of the chlorophyll present there. So, conclusion is chlorophyll is extremely necessary for photosynthesis. I hope you have understood this experiment.